Our today's topic is about protein metabolism. As in our previous video, we have learned about the deamination process, and its link I will give you in the description box. And let's begin about the transamination. And what is the process of transamination? It is the process in which the transfer of amino group from the amino acid to the keto acid, in which the amino group is transferred from the amino acid to the keto acid. So here is not removal of the amino acid, here is only the transformation of the amino acid. So here it is the amino acid, and this is the acceptor, this is the donor. So it is donating its amino group to the keto acid, and keto acid is accepting the amino group from the amino acid, and here the enzyme transaminase is playing role, and we can also say it aminotransferase. Okay, both are the same. And here when the amino acid giving its amino group, so the pyridoxal phosphate enzyme is getting that amino group and giving it to the keto acid. Okay, so in this process, this is the reversible process. Okay, pyridoxal phosphate is converting into pyridoxamine phosphate, and again it is converting into pyridoxal phosphate. It is reversible. So here the keto acid, now it is forming a new amino acid, and the remaining keto acid is here. Okay, so in this way it is the reversible process. Okay, and not only the alpha amino acid is going in the transamination process, but also the delta amino acid is also going in the transamination process, which is the ornithine, which is the delta amino acid. And now this is the reversible reaction, as we know, and here is the no removal of the amino group, only the transfer of the amino group is here going on. And the keto acid which are forming here, it may be alpha glutarate, oxaloacetate and pyruvate, which is going into the Krebs cycle again. Through here they are going to the Krebs cycle, and this process is going in the liver, kidney and brain and the heart, all of the tissues, and in the cytosol, in the tissue it is going in the cytosol and the mitochondria. All the amino acids are doing the transamination process except lysine, proline, threonine and hydroxyproline. Okay, then keto acid, this keto acid, what will they do? So they provide energy, or they can form glucose or ketone bodies of fat or non-essential amino acid. So this process is going on in the essential amino acid, which is dietary protein. Okay, which amino acids we are taking in our diet, they are doing this transamination process, and this reaction will get clear here. Now first of all we take example like alanine. So here the alanine is broken down, and alanine is an amino acid which is by breaking itself, and it is giving its amino group to the alpha ketoglutarate, and this alpha ketoglutarate is now forming glutamate. Okay, glutamate is forming here, and the remaining keto acid is pyruvate. So here the important enzyme is ALT, or we can say SGPT, both are the same, and PLP is a coenzyme which is playing role here. So the ALT has the function to breaking down the alanine and forming the glutamate. So here is the main thing is here is to synthesis of glutamate amino acid is synthesized here. And PLP is a vitamin B6 derivative enzyme. Pyridoxal phosphate is its full form. And now coming to the second example. So here enzyme AST, or we can say SGOT, is playing role, and coenzyme PLP is also present here. Here the glutamate is breaking down and forming aspartate. It is opposite to it, that here is the synthesis of the glutamate, here is the breaking down of the glutamate. These all reactions are reversible. Okay, these all reactions are reversible, but the main aim is in here is to synthesize the glutamate, and here is the breaking down of the glutamate and synthesizing aspartate. So here the glutamate is breaking down, it's giving its amino group to the oxaloacetate, and oxaloacetate is forming aspartate, and here the remaining keto acid is alpha ketoglutarate. Okay, then again this will go into urea cycle. Then it will go to urea cycle. Okay, and now the as we know that protein has its end product is amino acid, and that amino acid after the process of deamination, that amino group is going to the urea cycle. It means it is forming urea, and that urea is excreting out with the help of urine, which is very toxic to collect in our body. That is why we do not collect it and we excrete it in the urine. Okay, so here the amino group is excreted out. It will go to the, first of all it will go to the urea cycle, and then it will excrete out as in our urea, in urine as urea. Okay. So, what are the full form of enzymes? ALT is alanine transferase, that is why it is playing role here in the alanine. And now here SGPT, serum glutamic pyruvic transaminase. So, here SGPT, G means glutamate and P means pyruvates. So, you can remember it as GP, glutamate and pyruvate. And AST, it is aspartate aminotransferase, that is why it is playing role in the aspartate metabolism. And here the SGOT, serum glutamic oxaloacetic transaminase, it means G for the glutamate and O for the oxaloacetate. Okay. Now, one more thing, that this is very important as the clinical basis, clinical testing, like whenever we have some disease in our liver, hepatic disease like jaundice or something else, or sometime we have any kind of cardiac disease, that time the testing for the SGPT and SGOT enzymes are done. And if first of all you have to know that SGPT is a specific enzyme and SGOT is a sensitive enzyme. So, little bit disease is there in our liver, so it push SGOT to increase its level in our body, because it is very sensitive enzyme. So, it easily get increase its level, but as it is not that much important, because it is sensitive, it will increase its level and without if here is a little bit disease, it get increase its level. But SGPT is very specific enzyme, so it will not get increase its level in very small amount of harmness in our liver. So, that is why if there is a very big harmful thing is going on in our liver, that time the SGPT increase its level, and it is very important to testing this enzyme. Okay, so that is why it is important. SGPT, SGOT is not that much important in comparison to SGPT, because it is sensitive and it is specific enzyme. So, that is all about protein metabolism and the transamination process. And if you like my video, hit the like button and subscribe my channel and share it with your friends and keep learning. And you can join the Telegram channel for the notes and the quiz. So, I will give its link in my description box, link of the Instagram and email ID and all. Okay, so check the description box and keep learning. Thank you for watching.